Здесь могу читать, да? Хорошо, Максим, спасибо. Я тогда отключаюсь и начинаю. Хорошо, спасибо. Good morning, ladies and gentlemen. Today's lecture is devoted to, again, to the inflammation. We described this process in the last lecture, to continue today. The second option, secondary recognition of non-self, antigens, processing, presentation of antigens. We understand that some of you had the classes before devoted to these topics, but I think that in any case, what my lecture will help you even if you studied this before. In the drawing that you see in this slide, you see purple antigen presenting cell. This is dendritic cell that interacts to the red CD4 naive T helper cell or T0, TH0. You see many 1, 2, 3, 4, 5 receptors on the surface of antigen presenting cell that interact to 5 receptors on the surface of T cell. In the central position, you see a circular antigenic peptide of yellow color that is presented by major histocompatibility complex type 2 protein of the green color of antigen presenting cell. This antigen is presented to T cell receptor of helper cell. Upper and lower, you see interaction of the so-called core receptors that are required to form immunological synapse that is required to initiate activation of T helper cell. Or what is this? Well, very quickly, let's repeat. Acquired immunity and antigens. Antigen is a substance that body identifies as a foreign and towards which immune response is mounted. Antigen has many properties, but in the lower part of the slide you see two properties that we will discuss, but exception for antigenicity, immunogenicity, very important characteristics of antigens, enough size, this molecule must be large. Protein antigen has to be more than 10,000 daltons, 10 kilodaltons, the molecular weight, and molecule has to be rigid. Molecule has to be foreign. It means antigenic molecule may be recognized by acquired immune system only if this antigen demonstrates so-called fingerprints of the foreign non-self genome. In this case, of course, the foreignness of the antigen would be enough to initiate an immune response. And antigenicity and immunogenicity. You know that antigenicity of antigen, what here antigen is shown in the right part of the slide, is the red star with the triangle antigenic determinants. Antigenicity, this is a capacity of antigen to react to the antigen binding sites of antibodies. What here you see is that molecule of antibody that has two antigen binding sites in the upper part of antibody, first and second, with right antigen binding site now binds to the antigenic determinant of antigen, and this is a reaction that is named as the antigenicity. Immunogenicity. Immunogenicity, this is a capacity of antigen of initiating immune response, acquired immune response. Immunogenicity, this is a result of the many reactions that finally make immune cells of specific immune cells of replicating, I mean T lymphocytes and B lymphocytes, and after some cycles of the replication, progeny cells of this initial activated by antigen cells will be matured finally and B lymphocytes will be matured to the plasma cells that will produce soluble antibodies. With soluble antibodies shown here, the purple molecule interacting to the antigen. You know that antigens may be complete or incomplete. Incomplete antigens are named haptens. Haptens are low molecular weight compounds, usually of non-proteinaceous origin. Haptens are antigenic. It means they may react to the antibodies, but they are not immunogenic. In the right part of the slide, you see formula of the penicillin. Penicillin, this is antibiotic, antibacterial compound of the fungal region that is antigenic for human immune system and is itself cannot initiate synthesis of antibodies, but if antibodies were performed before, penicillin may bind to the specific to the penicillin antibodies. Antibody is shown here as a purple Y-shaped molecule. Why haptens or non-complete or incomplete antigens are not immunogenic? Because haptens cannot activate helper T cells. This is due to inability of haptens to bind to the major histocompatibility complex proteins. You know that MHC, major histocompatibility complex proteins, are involved in the presentation of antigens. And haptens by themselves, they're too small to be presented in the MHC protein complex to this MHC protein. Second, why haptens are not immunogenic? Second option, haptens cannot activate B cells by themselves due to the haptens are univalent. It means what penicillin molecule has only one part of the molecule that may be recognized by antibody, not more. One, it means univalent, one unit of antigenicity is not enough to activate B cells. They require multiple contact to antigenic determinants of the same specificity. Third, haptens cannot stimulate a primary secondary immune response by themselves because if they are capable of activating T cells, of course, they could stimulate primary secondary immune response. Complete antigens. Complete antigens, in the contrast to haptens, are large molecules. Here you see yellow large molecule of protein that has the antigenic determinants on the surface of these bulbs, it's yellow color, that directly react to the antibodies. You see that complete antigen must be enough high, the molecular weight 10 kilodaltons or more, and with this molecule, if it's non-self, may stimulate induction of the immune response. In the upper central, upper right part of the slide, you see drawing consisting of two parts. Upper part demonstrates curves in the graphs of the immune response development. Vertical axis demonstrates level of antibodies, and horizontal line demonstrates time. Below this figure, you see cells. It means, what does it mean, immune response, in the sense of the accumulation of cells and produced by the cells of the antibodies? Initially, in the human organism, in the lymphoid system, occur very few T lymphocytes and B lymphocytes that carry receptors specific for the certain antigen. It means in the moment of the inoculation of antigen to the human organism, firstly in the life, this inoculation can cause immediate result, immediate response of immune system, but this response is initiated. How? Antigen becomes engulfed by the antigen-presenting cells, the macrophages or dendritic cells, and they present antigenic peptides of the antigen to the cells, to helper cells, for example. At the same time, B lymphocytes, capable of recognizing this antigen, recognize directly this antigen and start to proliferate. Well, this proliferation is shown in this figure. Initially, 2 lymphocytes are drawn on the scheme, and you see how do they replicate. And by the 5th day, number of cells really is higher than before. After that, cells accumulate their progeny, and the first line of the B lymphocytes is matured to the plasma cells with 1, 2, 3, 4 purple cells. These cells first secreting immunoglobulins or antibodies. Antibodies secreting first are immunoglobulins class M. They form the blue peak on the figure in the graph in the upper part of the slide. At the same time, other B lymphocytes continuously replicate, produce their progeny that finally also mature to the plasma cells, and these second line plasma cells produce immunoglobulin type G, immunoglobulin class G. Immunoglobulin G curve of this immunoglobulin curve that shows this immunoglobulin accumulation is red. Now, blue line demonstrates total number of immunoglobulins of both immunoglobulin M and immunoglobulin G. Third type of cells that replicate in this situation, they are cells that are transformed to the memory cells. Memory cells are shown here as green cells. Memory cells not mature to the plasma cells and stop their replication after some serial dividing. They stop their replication in the immature form, in the form of the B cells. This is a primary immune response. Development of the immunoglobulin synthesis and accumulation of memory cells after first inoculation of antigen to the human organism. What is the secondary immune response? This is a repeat immune response. If the same antigen in the future will enter to the organism of the patient who was immunized before by this antigen finds not a few cells in the immune organs, in the lymph nodes, liver, spleen, but they contact to many B lymphocytes that were accumulated as the memory cells after the primary immune response, and they're shown green here. These memory cells recognize antigen, and they after that switch on to the replication. They proliferate and also produce population of the early plasma cells that produce immunoglobulin M. Immunoglobulin M producing the primary secondary immune response is secreted during the beginning of the immunizing process, and the concentration of immunoglobulin M increased to the same levels. It means we don't see increasing in the secondary immune response of immunoglobulin synthesis, but you know that exception for first line, second line of the plasma cells is matured from B lymphocytes that continues to replicate many times. And with these plasma cells of the second line, they secrete immunoglobulin G, and concentration of this immunoglobulin G secreted in the secondary immune response is much higher than the level concentration that immunoglobulin G producing the primary immune response. And presence of immunoglobulin G of certain specificity interesting for us, or this presence of immunoglobulin in the blood is much longer than presence of immunoglobulin G in the primary immune response. What do you see? You see effect of the enhancing of the antibody response by the involvement of the process of the and activation of the memory cells. Of course, after secondary immune response, many cells of green color here, or these B lymphocytes, accumulate in tissues without differentiation to the plasma cells. They're also memory cells, and their concentration in fluid tissues becomes much higher than their concentration after primary immune response. Of course, if the same host patient will be exposed to the same antigen in the future, this would be third immunization. All the things would be repeated, and the concentration of immunoglobulin would be higher than after the previous immune response. It means multiple immunizations of the humans or animals. Multiple immunization is named as hyperimmunization, and any new episode of initiation gives higher number of memory cells, higher number of the plasma cells, and higher level of immunoglobulin G. This is an immune response that demonstrates what is the immunogenicity. Of course, specificity is typical for complete and incomplete antigen specificity. Not specificity, I mean say about antigenicity. Antigenicity is capacity of immunoglobulins specifically react to the antigen, and immunogenicity, antigenicity is typical for both complete antigen shown here and incomplete antigen that you saw before. Complete antigens are large. Incomplete haptens are small and usually are not proteins. But if haptens are chemically combined to the large antigenic peptide or protein molecule, this covalently bounded to the protein haptens may serve as antigenic determinants of this large molecule, and in this case, haptens transform to the complete antigen. It means complete antigen may be produced if carrier protein of this large yellow protein is carrier protein, is chemically bounded, covalently bounded to the haptens, and this covalently bounded haptens on the surface of the antigenic peptide or antigenic protein serve as the antigenic determinant, means transform to the complete antigen. What in this case, what may be happen? For example, normal plasma proteins or cellular proteins of our organism may bind to certain types of haptens that circulate in our blood, and in this way, haptens may be transformed to the complete antigens and start to initiate immune response. And as a result, antibodies will accumulate in tissues, and these antibodies will react to haptens in the free form or haptens in the form bounded to the carrier protein. Antigenic determinants or epitopes. Epitopes are the small units of antigenicity. Now, for example, in the drawing, the left part of our slide, you see 1, 2, 3, 4 yellow antigenic structures that have epitopes of different specificity. What left yellow antigen has around origin epitopes. The next yellow antigen has triangle epitopes. The next squared epitopes. And every epitope that is unique reacts only to one type of antibodies. Antibodies are shown as the blue molecules. Every antibody shown here has three ends, tails. Upper tails 1 and second, they have antigenic binding sites, and these sites recognize specifically only one antigenic epitope. What's spherical epitope is recognized by left antibody, some triangle by the middle antibody, and squared by the right antibody. Epitopes are the smallest units of antigenicity. They are small chemical groups on the antigen molecule which can elicit and react antibody. In the right part of the slide, upper part, you see theoretical structure of virus. You see a red square labeling the spherical spike on the surface of this virus. And chemical analysis of the spike demonstrates in the right part of this figure tertiary structure of the brown amino acid chain in the tertiary structure of the protein. This protein in different parts of the amino acid chain has different state by different colors of different form epitopes. Of course, antigen that you see here is polyvalent because you see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 different epitopes. Each of that may react to the antibody. It means what this protein shown here may react to more than 10 different antibodies with a different specificity. Of course, this is a polyvalent antigen. One epitope, this is the one unit that may react only to one specific antibody. In the lower part of the slide, you see how does direct antibody to epitope. Epitope is shown in the round field of view as a yellow fragment of yellow molecule. You see that this yellow epitope consists of the antigenic epitope consists of the spherical or non-spherical atoms of the different chemical elements, nitrogen, carbon, hydrogen, or other. Of course, combination of these atoms, if it's rigid, forms the rigid structure of the epitope. Epitope may be recognized by antigen binding site of antibody. In the left part of this lower fragment of the slide, you see antibody consisting of 3 arms. 2 upper arms have the antigen binding sites, one site and other, second. Every site consists of the 2 chains, variable, labeled by letter V, variable chains of the polypeptides, purple polypeptide, heavy chain, and green polypeptide, light chain. Well, these chains form the antigen binding site, and this is a pocket with a specific configuration of internal atoms. And you see in the circle field of view that antigenic peptide of the yellow color contacts to the purple atoms and green atoms of the 2 chains that form the specific pocket with a specific internal configuration of the atoms that fit to the epitope. If internal configuration of the atoms of the antigen binding site with a high complementarity fits to the structure of the epitope, non-covalent bridges between atoms of epitope and atoms of the antibody establish really strong interaction. And this interaction is characterized by high specificity and strength. If structure of the antigen binding site wouldn't fit to structure of the epitope, interaction between atoms of to these molecules would be impossible, and recognizing of antigen would be absent. Some words about bacterial antigens. Bacterial antigens traditionally are named by the following letters. H antigens are antigens of the flagella, of the protein flagellum. What you see, bacterial cell, and upper bacteria has one flagellum, a lower bacteria has many flagella. H antigens are antigens of flagellum. Second type of antigens are antigens of pili, for example, F antigens. F antigens occur in the pili. Pili are not flagella, it means they are not involved in the movement. Pili are used by bacteria for the attaching to the other bacteria or human cells or the substrates. They are F antigens. Third type of antigens, O antigens. O antigens are antigens of the cell wall of the gram-negative bacteria. O antigens are antigens of the lipopolysaccharide, side chains of lipopolysaccharide. In the low drawing, O antigens are shown as the brown balls that occur below the purple capsular material. Capsular antigens are K antigens. They're shown as the purple material that overlaps all the bacterium and may hide O antigens below. Traditionally, capsular antigens of Salmonella typhi is named as a K antigen. Is named as a V antigen. V, capital and I, small I, V. This is a symbol characterizing that this antigen is associated to the virulence of bacteria. Virulence is capacity of causing disease. It means all the bacteria have K antigens, capsular Salmonella V antigen is analog of K antigen of the other bacteria. Bacterial antigens are recognized by specific antibodies. These antibodies shown here is the blue white shaped molecules. These blue molecules bind to the different antigens of the bacterial cell, to the O antigen, so V antigen or K antigen. And here you see three processes that initiated to destroy bacteria. In the upper right part of the slide, you see that antibody, blue bounded to the bacterial surface, reacts to the star-like C1 protein of the complement system. C1 protein forms the antigen-antibody complex to this antibody bound to the surface of bacteria. After that, C1 protein activates other C2, C4, C3, and other molecules of the complement system. And finally, C9 molecules. They are shown as the green rods. These green rods, C9 molecules, involved in the formation of the membrane attack complex. Here, membrane top complex MAC shown as the tube consisting of the C9 proteins, and this membrane attack complex causes penetration of the lipid membranes of the bacterium, and finally leakage of the liquid from the bacterium, losing of the ions, removing, and bacterium dies. This effect is named complement-mediated bacteriolysis. Lower, you see a blue cell. This is a neutrophil. Neutrophil may bind to the complement molecule that before was bounded to the surface of the bacteria. Usually, such the molecule, if not the C9 that may bind to the bacterium, is a C3b molecule, result of actination of C3 fraction of the complement. We see that this neutrophil has the orange C3 receptor for the C3b component molecule, and the bacterium becomes visible for this neutrophil due to the presence on the surface of the bacteria of the C3b molecules that are specifically recognized by C receptors, and they attract neutrophil to the bacteria. Neutrophil engulf this bacteria after that. And in the left angle of the slide, you see yellow macrophage. This is antigen-presenting cell that also recognizes bacteria via contact to the antibodies that bind to the bacteria. Macrophage here has the Fc receptors, green color receptor, to the Fc fragment of the antibody that bounded to the bacteria. What do you see? You see that antibody that labels surface of bacteria attracts macrophage and enhance the capacity, phagocytotic capacity of macrophage. Neutrophil also is attracted to the bacterium via recognizing of the complement molecule absorbed to this bacterium. Complement molecule C3b facilitates engulfment of bacteria by phagocytotic cell. And immunoglobulin G also facilitates engulfment of bacteria by phagocytotic cell. What effect of enhancing of phagocytosis by molecule absorbed to the surface of bacteria is named opsonization. Immune molecules that cause opsonization, immunoglobulins G or C3b component of the activated complement, are named opsonins. What the effect of opsonization is shown here. Of course, human and animal cells have many different antigens on the surface. These antigens now are labeled by the symbol CD and certain number. Some are surface antigens of the immune cells, and not only immune and non-immune cells may be marked by symbols differ from CD. For example, what here you see on the surface of B lymphocyte, white-shaped antibody molecule anchored to the membrane of this B lymphocyte by Fc fragment. This is a so-called B cell receptor for antigen, or immunoglobulin M or immunoglobulin D receptor for immunoglobulin receptor for antigen. This receptor is not named as the CD. This is a B cell receptor. Also, on the surface of B lymphocyte, you see a molecule of the MHC2, major histocompatibility complex, protein type 2, involved in the presentation of antigen. What in the lower part you see Fc gamma RI or CD32, what any symbol may be used. This is a receptor for Fc receptor of the Fc portion of the immunoglobulin class G. So CD receptors and other receptors identified on the surface of our immune cells, and that serve as the markers of specificity and differentiation of these cells. This is surface antigens that may be detected by recognizing of these human receptors by antibodies of animals that were immunized by human antigens. And CD receptors, non-CD receptors shown here, they are antigens for animals and animal monoclonal antibodies with the high specificity react to these receptors, identifying cells, the basis of the finding of the specific receptors typical for these cells. What are the specific receptors typical for lymphocytes? What in the right half you see B cell, in the left T cell. For example, only T cells may carry CD4 or CD8 receptors. CD4 and 8 are distinctive receptors of T cells. CD4 receptor is a distinctive receptor of T helper cells, of all the variants of T helper cells, all the populations. CD8 receptor, this is a receptor of the T killer or cytotoxic T lymphocytes. All T cells, independently helpers or killers, or cytotoxic, they have T cell receptor. T cell receptor, this is a receptor for antigen. This receptor is intimately bound to the CD3 receptor. CD3 receptor is typical also for T cells, for both helpers and killers. TCR recognizes antigen, antigenic peptide. If this recognition is happened, information of this information is transferred to the CD3 receptor, and from CD3 receptor, by the special signaling pathway, information is translated to the nucleus of T cell and initiates certain genes that are expressed and produce proteins that are markers and activation of T cells and participate in this activation. Exception for this, T cells have CD2 receptors, CD28, CD5, CD7 receptor. Receptors of B lymphocytes are from CD19 to CD22, CD19, 20, 21, and 22. Immunoglobulin antibody receptor of B cell receptor. CD40 receptors, MHC2 molecules, CD5 that are common for T lymphocytes and B lymphocytes, some of the B lymphocytes, and many other receptors. Of course, both the cells and all nucleated cells have MHC1 receptors on the surface. And because T and B cells are nucleated, MHC1 receptors must occur on the surface of these cells. Now, again, with the more details, B cell has immunoglobulin B cell receptor, antigen recognizing. MHC1 protein, what's shown here is the green at the top of the figure, MHC1 protein. B lymphocytes have MHC2 proteins that participate in the antigen presentation to the T helper cells when B cell combines antigen peptide of the microbe from the complex of these microbes to the MHC2 receptor, and in this way displace this antigen MHC2 molecule complex to the T helper cell, naive T helper cell. After this antigen presentation, antigen presenting cells have to make contact between antigen presenting cell, what in our this case B lymphocyte, and the T helper cell more stronger and more rigid. But in these interactions, very important role of the core receptor, for example, CD40 receptor of B lymphocyte or dendritic cell or macrophage binds to the CD40 ligand to helper cells. This is additional core receptor recognition of cells. Here you see CR1 and CR2 complement recognizing receptors. CDR also is named the CD35 and CDR2 is the CD21. Both react to the C3b portion of the complement molecule. T helper cells shown here. They have CD4 receptors, other CD8. If are they immature yet, they may have both these receptors and are named double positive cells. Double positive are precursors of CD4 or CD8. So, T cell receptor recognizes foreign microbial antigen or virus antigen foreign, and the C3 receptor is the receptor that is activated after recognizing of antigen by T cell receptor. Here you see green MHC1 protein. MHC1 protein, this is a protein that presents endogenous microbial proteins were complex into the MHC1 that will display this endogenous virus or tumor proteins of this T helper cell to the T killer cell. T helper cells, of course, may be infected by the different agents, for example, the human immunodeficiency virus, HIV, and antigens of this virus are presented on the surface of virus-infected T helper cell in the complex to MHC1 protein for recognizing this complex by T killer cell. A T killer cell is CD8 cell. Immune responses may be subclassified to different forms. One of the classifications divides them to the humoral, acquired immune response, antibody-mediated, and cell-mediated immune response. Acquired cell-mediated immune response. Antigens initially interact with antigen-presenting cells after injected to the human body. Antigen-presenting cells are phagocytotic cells such as macrophage and dendritic cells, and B cells. They are also antigen-presenting cells. Antigen, after engulfment by the antigen-presenting cell, after processing, is exposed on the surface of the antigen-presenting cell in the complex with the MHC2 molecule. And in this form, this antigen may be presented to the T helper cell that recognizes antigen MHC2 complex by the CD4 receptor of the T helper cell and T cell receptor recognizing antigen. T helper cell replicates and undergoes to the maturation, to the finally matured, completely matured T helpers type 1, T helpers type 2, T helpers type 9, helpers type 17, T follicular helpers, and T regulatory helpers. It means there are many subpopulations of helpers that differ one from other due to the synthesis of different cytokines that have different biological effects. If T helper cell produces T helper cell type 2, interleukin 4, interleukin 5, this signal via secretion of these interleukins, stimulation signals, as shown here by the plus in the green field, stimulates B cell. B cell, if before recognizes by the immunoglobulin receptor antigen, and after that is stimulated by the humoral signal of the helper cell in the form of the CD4 and 5, these B cells start proliferation, and majority of these cells are transformed to the finally completely matured form of the B cells that is named plasma cell. Plasma cells stop their replication, live only 2 days, but during these days produce very high amounts of the antibodies. The appearance of the high amounts of antibodies in the human organism after contact with the antigen is named humoral antibody-mediated immune response. Some B cells, they stop their replication and serve in the future the B memory cells. T helper cells. T helper cells, after stimulation by the antigen, also replicate, also mature to the finally matured effector forms, some helper type 1, 2, and other. And some of the cells only also transform to the T memory cells. Cytotoxic T cells. They are important part of the cell-mediated immune response. Cytotoxic T cells recognize antigens after endogenous presentation of these antigens in the complex to the MHC protein type 1, major histocompatibility complex protein type 1. In this case, cytotoxic T cell may recognize foreign antigens in the complex to MHC1 and the surface antigen presenting cells if antigen presenting cells are infected. If they're not infected, cytotoxic cell may be stimulated by some cytokines of the antigen presenting cells that is secreted after their stimulation. For example, interleukin 12 of antigen presenting cell stimulates naive CD8 T cell to be transformed to the cytotoxic cells. And helper T cells, via production of the interferon gamma, for example, also may stimulate cytotoxic T cells for the killing of the target cells infected by viruses or bacteria. So, antibody synthesis, this is a result of the humoral acquired immune response, involves interaction of at least 3 types of cells: antigen presenting cell, helper T cell, and B lymphocyte. Antigen presenting cell engulfs microbe. Processed antigen, it means destroys microbe in the phagolysosome and isolates from this microbial organism epitopes, antigenic epitopes. They're shown with the yellow triangles. They are combined to the MHC2 proteins and expressed on the surface of the antigen presenting cell, this dendritic, for example, macrophage, to the T helper cells. T helper cells, by the T cell receptors, recognize specifically antigenic peptide if this peptide fits to this real T helper cell, and by the T helper cell, while the CD4 molecule recognizes self nature of the MHC2 protein. And if this is a human MHC protein, human T helper cell, and individuals are genetically similar, that if this is a helper and a macrophage of the same origin, of the same organism, say host T helper cell becomes activated and produces cytokines. If this is a T helper type 2, the cytokines is interleukin 4 and 5. These cytokines stimulate B cell that before was stimulated by other signals. So B cell directly via B cell receptor for antigen, immunoglobulin receptor, recognizes specific antigen on the surface of bacteria or in the soluble form. First signal stimulation. After that, B cell is stimulated by the cytokine interleukin 4, for example, produced by T helper cell. And third stimulation, B cell is co-stimulated by interaction of the CD40 receptors of B cell to the CD40 ligand receptor of the Th2 cells. Now, some other co-stimulating molecules also are involved. Well, this is a simple scheme of the interaction of cells in the humoral immune response. We know that immune response, humoral immune response, antibody immune response may occur in 2 forms: the T-independent immune response or T-dependent immune response. T-independent immune response means that activation of B cells and synthesis by these cells and plasma cells that are transformed from these B cells, all this antibody synthesis may occur without presence of antigen-specific T helper cells. T-independent immune response is realized is the response for some of the carbohydrate antigens of the bacterial polysaccharides. What is happened? B cell directly, via B cell receptor, antibody receptor, binds to the carbohydrate chain of the bacterial polysaccharide, and binding of the many epitopes, same epitopes of the same structure of this carbohydrate, simultaneously react to many B cell receptors on the surface of B cell. Multiple irritation, activation is happening. In this case, without T helper cells, without cytokines of these cells, plasma cell replicates and produces immunoglobulins class M. Only immunoglobulins class M, not G. But for vast majority of the antigens, for example, antigenic peptides, for proteins, immune response must be T dependent. What is happening in this case? Antigen presenting cell with blue interacts to the glycoprotein. What blue is the peptide protein part and brown sugar part. This antigen presenting cell engulfs this antigen or bacterium having this antigen, destroys in the phagolysosome this antigen, isolates antigenic peptide from this structure of antigenic peptides, combine these peptides to the MHC histocompatibility complex type 2 MHC2 molecules, and the antigen peptide MHC2 molecule is exposed, is displayed for the T helper cell. T helper cell recognizes this antigen peptide by the T cell receptor, or this blue arm of the T cell receptor. After the T cell becomes activated. Of course, this is a CD4 T helper cell type 2, because we say about synthesis of antibodies. Activated T helper cell was shown in the lower part of the slide and matched by the red circle. Well, this activated T helper cell type 2 produces interleukin 4 and interleukin 5. In the same time, this T helper cell interacts to the peptide displayed by B cell. B cell may serve as an antigen presenting cell, and in this part of the slide labeled by the letter C, T helper cell interacts to B cell as to the antigen presenting cell. B cell, via the immunoglobulin receptors, binds to the antigen, and carbohydrate part of this antigen stimulates synthesis by B cells of the immunoglobulins M. But later, antigenic peptide recognized by T cell receptor and by the MHC2 receptor of B cell activates maturation of these B cells to the plasma cells, and plasma cells of this second line produce second type of immunoglobulin, immunoglobulin G. We told you about different subclasses of T helper cells. All these helper cells has CD4 positive cells. Today, at least 1, 2, 3, 4, 5, 6 subpopulations of T helper cells have been described. T helper type 1, secreting interferon gamma, interleukin 2, and tumor necrosis factor alpha and beta. This T helper type 1 cell maintains cellular immune response directed against viruses, intercellular bacteria that serve as intercellular parasites, and intercellular protozoa and fungal parasites. It means T helper type 1 response is directly to kill all cells infected by these intracellular parasites. T helper type 1 secretes interferon gamma, and this interferon gamma serves as the immunomodulator. It means interferon gamma, when it's present in the microenvironment where naive T helper start be activated, under the control of interferon gamma, stimulation of interferon gamma, naive T helper cell, T helper 0, is matured to the T helper type 1. It means T helper type 1 produces interferon gamma, and interferon gamma stimulates accumulation of T helpers even type 1. At the same time, interferon gamma inhibits formation of T helpers type 2 from the naive T helper cells, and sort of the antagonist of the T helper type 2 synthesis and maturation. T helper type 2 is responsible for antibody, humoral immune response. T helper type 2 secretes interleukin 4, 5, 10, and 13. Interleukin 4, this is also immunomodulator. Interleukin 4 stimulates maturation of naive T helper cells into the T helper cells type 2. And at the same time, interleukin 4 inhibits maturation of naive T helper cells to the population of T helper type 1. It means T helper 1 disturbs accumulation of T helper type 2 cells. Interleukin 4 disturbs accumulation of T helper type 1 cells. Of course, these inhibitory effects are not absolute and can completely prevent maturation to the helper 1 or helper 2 subpopulations. But proportions between matured helpers may be controlled by these immunomodulating cytokines. Exception for helper type 1 and 2, T helper type 9 have been described. This is a producer of interleukin 9 that play a role in the protection against helminths, stimulating anaphylactic allergy reaction. The next, T helper 17. T helper 17, this is a helper, matured form of that, produces interleukin 17A, interleukin 17F, and interleukin 22, and exception for this interleukin 21, 25, 26, and tumor necrosis factor. Of course, cytokines shown by the red color are prevalent. T helper 17, via synthesis of these cytokines, stimulates immune response by the recruitment of the neutrophils from the vascular space, from the vessels to the infected tissues. The T helper 17 from one side stimulates antibody immune response, collaborating to helper type 2, from other influx to the infected zone of the organism of neutrophils, causing neutrophil infiltration of tissues and damaging of the microbes and human tissues affected by these neutrophils. T follicular helper produces cytokines that usually produced by other type of helpers. It means T helper, follicular helper produces interleukin 21 that typically is produced by helper 17. T follicular helper secretes interferon gamma typical for helper type 1 and interleukin 4 typical for helper type 2. It means T follicular helper produces cocktail of cytokines that stimulate replication of all other T cells, including helper 1, 2, 17, in the lymphoid organs. Follicular, it means T follicular helper stimulates proliferation of activated T cells in the germination follicles of immune organs. And last, T-regulatory cell. T-regulatory cell, CD4 positive T-regulatory lymphocyte. This lymphocyte produces two types of cytokines, transforming growth factor beta and interleukin 10. Both cytokines are immunosuppressive. Cocktail of both these cytokines causes inhibition of replication of precursors of all other helper cells and causes lowering of the concentration of mature T helpers, causing their apoptotic death. It means T-regulatory helper cell causes down-regulation of immune responses of different type and prevents uncontrolled destroying of own tissues by the inflammation stimulated by T helpers type 2 and helpers type 17. Well, this is a brief review of the different types of helpers. Some words about terms. Cytokines. Cytokines are small peptides, molecular weight from 5 to 20 kilodalton. They're important in cell signaling. It means interaction with different types of cells. Cytokines in the cell signaling as the immunomodulator agents. Not signaling. Interferon gamma produced by T helper type 1 creates signal inhibiting maturation of naive cells to the T helpers type 0, naive T helper cells. Interleukin 4 of the T helpers type 2 prevents maturation of T helpers type 1 to the matured form and prevents synthesis of interferon gamma and inflammation. So cytokines induce, consist of chemokines, interferons, interleukins, tumor necrosis factors, lymphokines, and some growth factors. It means cytokines, this is all the variety of these biologically active molecules. Cytokines are produced by different types of cells, T, B lymphocyte, mast cells, and so on. Cytokines act through cell surface receptors. It means cytokines cannot cross the lipid bilayer of the lipid membranes of the human cells. Only protein receptors may react to the presence of cytokines, transmitting signal from the surface of cell to the cytoplasm and to the nucleus with the activation of this cell. Cytokines modulate the balance between humoral and cellular immune response. Interferon gamma produced by helper type 1 stimulates cellular immune response. Interleukin 4 produced by T helper type 2 mostly stimulates humoral immune response. Cytokines regulate maturation, growth, and activation of particular type of cells. Some cytokines enhance or inhibit action of other cytokines. In this slide, you see brief characteristics of 4 cytokines. Upper tumor necrosis factor interferon gamma are cytokines that enhance cellular immune response or immune response type 1 or T helper type 1 response. First is tumor necrosis factor alpha. Is produced by many types of cells, including macrophages, mast cells, lymphoid, and endothelial cells and fibroblasts. Effects. Tumor necrosis factor alpha is a pro-inflammatory cytokine that activates fever and septic shock with concentration of this cytokine in blood is high. Tumor necrosis factor alpha stimulates synthesis of acute phase proteins in the liver. For example, C-reactive protein in the liver that inhibits spread of the bacteria from affected zone to other zones of human organism. Tumor necrosis factor stimulates phagocytotic reactions and synthesis of interleukin 1 by macrophages. And this is a chemoattractant for neutrophils. Interferon gamma, second pro-inflammatory cytokine, is produced by T helper type 1 activating T killer cells or CD8 cytotoxic lymphocytes and activated natural killer cells. Interferon hub activates macrophages and natural killer cells and induce expression of MHC2 and MHC1 proteins on the surface of cells. It's primary cytokine that defines T helper type 1 cells. It means this is a primary cytokine stimulating growth or maturation of T helper type 1. Second two cytokines in the lower part of the slides are named cytokines that enhance antibody response, or type 2 cytokines, or Th2 response. Interleukin 4. Interleukin 4 is secreted by T helper type 2, mast cells, eosinophils, and basophils. The major producer, of course, is interleukin, is T helper type 2. Effects, biological effects. Interleukin 4 is anti-inflammatory cytokine. It induces differentiation of naive T helper cells, or T helper 0, to T helper cells type 2. It induces B cells differentiation into plasma cells and B cells class switching to immunoglobulin E. It means T helper type 2 stimulates replication of B cells activated and transformation of their progeny to the plasma cells with the producing of antibodies. Among antibodies, interleukin type 4 maintains more actively than other synthesis of immunoglobulin E. We have to understand and remember that immunoglobulin E, the major immunoglobulin, participating in the development of allergy reactions or hypersensitivity reaction. It means interleukin 4 may have negative effect in some people. Interleukin 4 decreases the production of T helper 1. Decreases activation of macrophages, decreases synthesis of interferon gamma and interleukin 12 by dendritic cells. And interleukin 10. This is an interleukin produced by monocytes, T helpers type 2, mast cells, but CD4, CD25, FOXP3, regulatory T cells. It means interleukin 10. This is one of the major cytokines produced by T regulatory helper cells. And this also is produced by activated B cells. Interleukin 10 is anti-inflammatory cytokine. It deregulates the expression, down-regulate, decreases the expression of T helper type 1 cytokines. Tumor necrosis factor, interferon gamma, and other. Interleukin 10 decreases synthesis of MHC2 molecules by the antigen-presenting cells and decreases synthesis of costimulatory molecules in macrophages. Interleukin 10 enhances B cell proliferation and antibody production, but in principle serves as the immunosuppressing agent. In the process of the humoral immune response, B cell activation is happening. One of the topics of our lecture is development of humoral immune response to antigen presentation. B cell is one of the antigen presenting cell, and at the same time is the cell producing antibodies. So B cell is the central cell of antibody synthesis, where B cell immunoglobulin receptors react to the antigens. Some of these antigens are engulfed by B cells, fragmented to the peptides, and B cell presents on the surface antigenic peptide with this blue in the complex with the MHC molecule class 2. Stimulated by recognizing of antigen, B cell produces additional amounts of the MHC proteins class 2 on the surface, and co-receptors that are named CD40 and some other co-receptors. After that, co-stimulation of B cell and co-stimulation of the T helper cell is happened. Co-stimulation, additional stimulation. B cell presenting antigenic peptide with the complex to MHC2 proteins present this peptide to the T helper cell type 2 or T helper cell naive that would be transformed to the type 2 in the future. T helper cell via T cell receptor recognizes antigenic peptide shown blue. CD4 receptor of the T helper cell recognizes MHC receptor type 2 is itself to confirm that T helper and B cell may interact one to other. Because you remember that major histocompatibility complex proteins are proteins that are markers of self or non-self. Only if this MHC2 self, it means producing the organism of our patient, and if T helper also is producing the organism of our patient, CD4 interacts with this MHC2, confirming that this is interaction is legitimate, is correct interaction. So what is happening? T helper is activated additionally by this interaction, and B lymphocyte by this interaction. Of course, core receptors, CD40 receptor of B cell, interact with the and bind to the CD40 ligand of the T helper. You see, TCR finds the peptide, recognizes peptide, and CD40 ligand of T helper finds and recognizes CD40 ligand of B lymphocyte. What is happened after that? After that, T helper cell becomes activated to synthesis of corresponding cytokines. These cytokines, if this is T helper type 2, in the antibody synthesis is interleukin 4, interleukin 5. These cytokines react to the receptors for interleukin 4 and interleukin 5 on the surface of B lymphocyte. We know that this B lymphocyte was activated before by antigen recognizing by the interaction of core receptors CD40 to CD40 ligand, and was stimulated by these cytokines. It means 3 signals. These 3 signals make B lymphocyte replicating, and finally transforming of the progeny of the cells to the plasma cells producing antibodies of this region that fit for this antigen, and of the memory B cells. Antigen presenting cells may be subdivided to professional antigen presenting cells and atypical antigen presenting cells. Professional antigen presenting cells, dendritic cells, macrophages, and B cells. They produce MHC class 2 proteins constitutively. Constitutively means MHC2 molecule occurs on the surface of the cells in any moment of their life, independently of the presence of antigen. Professional antigens express co-stimulatory molecules following activation. It means what? CD40 ligand would be synthesized by the CD40 receptor would be synthesized by B lymphocyte, the antigen presenting cell, only after stimulation of B lymphocyte by antigen. MHC2 type, MHC protein type 2 would be expressed on the B lymphocyte in any moment of the life. It means constitutively, it means occurs in the every time. In the contrast, atypical antigen-presenting cells induce expression of MHC class II molecules only after activation by certain cytokines or by the presence of the pumps, dumps, typical for microbes. Dendritic cell is a unique antigen-presenting cell capable of activating naive T helper cells. Capacity of the antigen presentation of these cells is higher than capacity of all other antigen-presenting cells. Dendritic cell primarily recognizes foreign molecules as the pathogen-associated molecular patterns that are recognized by toll-like receptors of the antigen-presenting cell, including dendritic cell. Signaling about this recognition activates nuclear factor kappa B, and nuclear factor kappa B activates expression, it means transcription and translation, finally synthesis of by the antigen presenting cell of such substances as the co-stimulatory and adhesion molecules. So it means synthesis of CD40, CD80, and CD86 co-stimulator molecules in the cell is intensified. Additionally, this recognizing of the microbial pathogen-associated molecular pattern causes activation of synthesis of such cytokines as the CCL2 chemokine and interleukin 12. CCL2 interleukin 12 stimulate replication of different types of the helper cells. Additionally, antigen-presenting cell as antigen-presenting cell engulfs bacterium, destroys this bacterium, releases, isolates from this bacterium, uptakes of this bacterium antigenic peptides. This process is named beginning of processing. Antigenic peptide is combined to the MHC2 molecule, what here, the green molecule, that is formed in the endoplasmic reticulum and completely matured in the Golgi apparate. Matured MHC2 protein in the complex to antigenic exogenous peptide of bacterium is exposed on the surface of the dendritic cell. This is a event that is named presentation of antigen. It means exogenous microbial antigen is presented in the complex of MHC class II molecule. Naive T helper cell, by its T cell receptor, specifically react to the antigenic peptide. Only T helper cells that specifically recognize this peptide may be activated. If vast majority of the cells, they have different specificity and can be activated by this antigen, but few cells, yes, would be activated. To confirm legitimate character of the interaction of this antigenic stimulation of T helper cell, CD4 receptor of T helper cell binds to the MHC2 molecule of the antigen presenting cell, and in this way allow reaction of the T helper cell to the recognizing of the antigen by T helper cell. Dendritic cells antigens, dendritic cells present antigens shown with this green to naive T helper cells that finally stimulate T cells and polarization. T helper type 1, T helper type 2 polarization. It means naive T helper cell may be polarized, it means matured, to different types of the T helpers. What in this figure you see polarization for T helper type 1, activating cellular immune response by synthesis of interleukin gamma and tumor necrosis factor beta. And this naive helper may be polarized to helper type 2, producing interleukin 4, 5, 13, interleukin 10. How these signals are realized? Three types of signals activate naive T helper cell. First signal, this is a signal from interaction of T cell receptor, specific interaction to the antigenic microbial peptide presented by MHC molecule exposed on the surface of antigen presenting cell. First signal, recognition of antigen. Second signal, signal generated by contact of co-receptors. CD28 receptor at this moment interacts to the CD80 and CD86 receptor on the surface of the antigen presenting cell. And the second core receptor interaction, CD40 ligand receptor of the naive T helper cell interact to the CD40 receptor of the antigen presenting cell. Second signal, co-stimulatory signal. And the third signal is humoral signal. When activated, antigen presenting cell secretes either interleukin 12, either CCL2 chemokine. If antigen presenting cell mostly with this, due to the interaction to different pumps, to different cytokines, if finally this dendritic cell, antigen presenting cell, will produce interleukin 12, this interleukin 12 will bind to the interleukin 12 receptor of the T helper cell, and the signal, third signal, humoral signal, will initiate polarization of naive T helper cell to the T helper type 1. Other situation, if dendritic cell mostly will secrete CCL2 chemokine, this chemokine will react to the corresponding receptor of the T helper cell, and this interaction will stimulate polarization of naive T helper cell to the T helper type 2. So, polarizing signal number 1. This is an antigen recognition by T helper naive T helper cell. So, antigen presenting cell, shown purple, displays on the surface T helper MHC class 2 molecule that contains in the antigenic peptide binding cleft antigenic peptide. This is an exogenous peptide, microbial peptide. This peptide is recognized by antigenic binding site of the T cell receptor, and this interaction is confirmed by reaction of the CD4 receptor of T helper cell to the MHC2 molecule of the antigen presenting cell. Well, this last interaction is not shown. This is the first polarizing signal. In this signal, very important role plays T cell receptors. T cell receptors, of these red T cell receptors, of the T cells may be of alpha beta or gamma delta type. Alpha beta are heterodimers that are receptors of majority of T helper cells and T killer cells. Alpha beta T cell receptors recognize the antigen by the antigen binding cleft between first and second alpha and beta chains of these receptors. Only if this T cell receptor recognizes antigen, only the complex with the MHC class 1 or MHC class 2 molecules. T cell receptor of the T cells itself doesn't process signaling domain. It means directly can transform activation signal, transport the signal to the cytoplasmic molecules of the signaling pathways. Due to this, T cell receptor deliver the signal of activation to the CD3 molecules. CD3 molecules may be of the epsilon gamma, epsilon delta variants, and these molecules that have transmembrane peptide fragments activate signaling pathway that changes the activity of the activating cell, of the T cell, stimulating maturation of the cell and activation of the cell. Gamma delta TCR, T cell receptors. These receptors occur in a small portion of T cells, of T helper cells and T killer cells. What here you see gamma delta T cell that has gamma delta TCR that will react to the endogenous peptide presented by the MH3 molecule type 1. Of course, this gamma delta TCR shown in this figure is T killer cell, CD8 cell, because MHC1 molecule may be recognized by CD8 receptor of this cell that is not shown here. And if target cell has MHC1, of course, brown T cell is killer. Gamma delta T cells have less T cell diversity compared to alpha beta. It means variants of antigens can be recognized by gamma delta TCR is lower than number of the variants that may be recognized by alpha beta. Gamma delta T cell receptors are enriched at epithelial structures, mostly present in the epithelial structure. Gamma delta T cell receptors appear to respond to self molecules that signal potential danger or cellular stress. It means mostly cells that have gamma delta receptors serve as killer cells. Gamma delta TCR, they have shown anti-infection, anti-tumor, and anti-tumor effect and antivirus effect. And they have immunoregulatory activity. So here you see effects of gamma delta T cells. They directly promote immune cells maturation and activation by triggering B cells. It means they activate triggering of the B cells, what here, and transforming of B cells to the plasma cells producing antibodies. Gamma delta T cells help dendritic cells maturation. What dendritic cells maturation from the precursors? Gamma delta T cells they activate priming of alpha beta T cells. It means they participate certain role in the activation of B cells. Gamma delta T cells regulate activity of the epithelial cells that involve in the immune response, microfold cells, for example. And they cause lysis of infected stress cells, it means tumor cells. In this slide, you see activation of T helper cells by the antigen presenting cell that presents antigenic peptide shown red to the MHC antigenic peptide shown red in the complex to the MHC molecules to the T helper cell. T helper cell via CD4 receptor recognizes MHC2 molecule itself, and in this case, the genic peptide is recognized by specific T cell receptor, and the formation is transformed, activate CD3 receptor that initiates activation of the cell, activation of the T helper cell. It means transforming T helper cell, replication of cell, transforming of progeny of the cells to the mature type of T cells, helper type 1 or 2 or other, dependence on the microenvironment and presence of the cytokines. This is an activation of T helper by the exogenous peptide. In the left part, you see activation of cytotoxic T cell by the presentation, endogenous presentation of the endogenous antigens. In this case, antigenic peptide of virus, infecting cell, or tumor is presented by the MHC1 protein. MHC1 protein is checked as a self-molecule by CD8 cell of the cytotoxic T cell, and the cytotoxic T cell becomes activated after this recognizing of antigenic peptide by activation of CD3 receptors. So, exogenous peptide presentation primitive scheme is shown here. In the endoplasmic reticulum, MHC2 molecule is synthesized and becomes hided by the clip peptide special that closes the antigen binding cleft of the MHC2 molecule. This is antigenic. This is a presentation of exogenous. Antigen means that exogenous pathogen, virus or bacterium, is engulfed by this dendritic cell or macrophage that is shown here. Microbes destroyed, antigenic peptides are isolated from this bacterium, and after that, in the Golgi apparate, they are injected to the area in the Golgi apparate where MHC2 protein is present. Exogenic peptide causes removing of clip, blocking peptide from the antigen binding cleft of the MHC2, and in this cleft, exogenous antigen is mounted, and in this form is exposed, transported to the surface. It occurs on the surface of the complex antigenic peptide present in the antigen binding cleft of the MHC2 class molecule. So, here you see two schemes of the presentation. On the right part, you see presentation of exogenous antigen. In the endoplasmic reticulum, MHC2 molecule is synthesized together with invariant chain shown yellow. Red MHC2 molecule after that migrates to the Golgi apparate, and in the Golgi apparate, invariant blocking chain is destroyed, and clip fragment of the chain remains in the place where antigen binding cleft of MHC2 molecules happen. Exogenous antigen, for example, fragment of the engulfed bacterium, parts of this bacterium is digested in the cell, parts of this peptides of the bacteria are cutted for the fragments for antigenic peptides. These antigenic peptides are inoculated to the Golgi complex part where MHC2 peptide is present. Antigenic bacterial peptides interact to the antigen binding cleft of the MHC2 molecule after removing of clip fragment, and in this form MHC2 class molecule together with antigenic peptide exposed on the surface. In the endogenous presentation, situation is different. Endogenous parasite, for example, virus, during replication inside of the cell, produces proteins. Some of these proteins appear in the cytoplasm of the virus-infected cell. This virus protein, together with damaged or maybe affected or changed proteins of this own cell, undergone to the destroyed with the proteasome. Proteasome destroys antigenic peptide for the fragments, short fragments of 10 or 20 amino acids. These fragments, through a special pouring system that is named TAP, are transported to the endoplasmic reticulum. In the endoplasmic reticulum, these antigenic peptides bind to the MHC1 molecule before and during process of maturation of this molecule, and MHC1 molecule with antigenic peptide from the endoplasmic reticulum is transported to the Golgi apparat, from the Golgi apparat to the surface, and exposed on the surface in the complex class 1 MHC molecule combined to the antigenic peptide. But green peptide is the peptide isolated from the products of the intercellular parasite that infect this cell. Here you see that antigen-presenting cell expose MHC2 molecule on the surface together with antigenic peptide. This peptide, when is recognized by T cell receptor of the T helper cell, causes activation of T cell if CD4 receptor of this helper will confirm that will react chemically to the MHC class molecule, recognize that this is a self molecule. Now, after that, parallelizing signal number 2 is happened. This is a signal of interaction of co-receptors. For example, B7 receptor of the antigen-presenting cell reacts to the CD28 receptor of antigen-presenting cell. This is after presentation of antigen to T cell receptor, of course. And CD4T receptor antigen-presenting cell interact to the CD40 ligand of the T helper cell. What do you see? You see formation of the immunological synapse that was initiated by antigenic presentation of the exogenous antigen from MHC. So in the complex of with MHC2 class molecule to the T cell receptor helper cell, and this interaction is enhanced and makes more rigid, done more rigid by additional interaction of the other ligands that stick helper cell to the antigen presenting cell. And third activating signal, this is polarizing signal number 3. This signal is maintained by cytokines. For example, when antigen presenting cell produces interleukin 12, this interleukin 12 stimulates maturation of naive T cell to the T helper type 1. If prevalent cytokine produced by antigen presenting cell is CCL2 chemokine, this chemokine reacting to the receptors, chemokine receptor of the T helper cell, stimulates transformation of the cell to the T helper type 2. This is a third cytokine mediated signal. Of course, in this maturation of naive T helper, very important role is the role of the cytokines that are produced in the same time by the mature T helper that occur near to this naive cell. For example, interferon gamma of the T helper type 1, of course, also establishes very powerful humoral signal that stimulates maturation of T helper type 1, T helper 0, T helper naive cell to the T helper type 1. It means here you see two variants of the cytokine stimulation of the maturation of naive T helper to the helper type 1. First signal, interleukin 12 of the antigen presenting cell. Second, interferon gamma of the T helper type 1. And oppositely, naive T helper cell would be stimulated to helper type 2 in the presence of the excess of interleukin 4, 5, interleukin 13, and of the CCL2 chemokines produced by different types of the cells trying to change character maturation of the naive T helper cell. Now, this slide we saw before, antibody synthesis is possible if antigen presenting cell activates T helper, and T helpers mature to helper type 2, where cytokine 4 and 5, this T helper and its cytokine stimulate B cell that before that was stimulated by antigen directly via the immunoglobulin receptor and co-stimulatory signals set online of the activation also important when CD40 ligand of helper to stimulate interact with the CD40 receptor of the B cell. Now, here you see scheme that we discussed before, endogenous antigen presentation, this presentation of endogenous parasite, and association of MHC1 protein to the peptide of this parasite in the Ralph's endoplasmic reticulum is the early event. In the contrast, binding of the exogenous peptide to MHC2 molecule this is a late event because it occurs after transportation of the MHC2 molecules to the Golgi complex, to the Golgi apparatus. Now here you see presentation that we discussed before. If endogenous antigen is presented, it means CD4 cell is involved in this interaction. What is happening in this case? Antigen presenting cell having intercellular parasites present epitopes of these parasites, antigenic peptides, in the complex to MHC1 molecule to the T cell receptor of the naive CD8 killer cell. This CD8 naive cell also may be naive before stimulation. If co-stimulatory signal is happened, this naive cell additionally acquires signal from the T helper type 1. T helper type 1, of course, is transformed from naive cell when antigen to the T helper is presented by antigenic peptide together with MHC class 2 and in the presence of co-stimulatory signals and in the presence of interferon gamma, CD4 naive cells are transformed to the CD4 helper type 1 cells, helpers type 1. Helpers type 1 produce interleukin 2 and interferon gamma. Interleukin 2 and interferon gamma stimulate activated naive CD8 killer cell, stimulated by the antigen recognizing and co-stimulatory signals, to the transformation, to the maturation, to the differentiated cytotoxic T lymphocytes. Differentiated cytotoxic T lymphocytes have granules, vesicles, with the cytocidal substances, granzymes and perforins. Granzymes and perforins are the toxic for the human cell substances accumulated in the granules of mature differentiated cytotoxic T lymphocytes. If these cytotoxic T lymphocytes will recognize target cell via recognizing of microbial endogenous antigen in the complex to the MHC class 1 protein by the TCR, it means if cytotoxic T cell, T killer cell, all by the TCR recognize antigen presented by the MHC1 molecule, and one MHC, this MHC1 molecule of the target virus infected cell would be recognized by CD8 receptor as self, this cytotoxic cell will understand that cell that is faced to this cell is own cell because he has the MHC1 that legitimately react to the CD8 receptor, and this own cell has the foreign antigen to that specific TCR receptor of this T killer cell. In this case, after activation of T killer cell stimulates movement of the granules to the area that contacts to the target cell. Cytoplasmic membranes of T killer cells and plasma and the lipid membranes of the granules contain enzymes and perforins are fused. Perforins are released to the cleft between cytotoxic cell and target cell and produce pathological pores in the membranes, lipid membranes of the target cell, the virus infected cell. Through these pores, perforins, the enzymes, hydrolytic enzymes, enter to the cytoplasm of the target cell and causes hydrolysis of proteins and nucleic acids of this cell, and additionally activate apoptosis of the cell, and target cell becomes killed. This attack is the result of the presentation of endogenous antigen by the target cell. Now here in this slide you see details. Yellow, this is a own virus infected cell or own target cell. Purple, this is a cytotoxic T lymphocyte. In the left part of the slide you see that TCR receptor of T killer cell interacts to the red antigenic peptide of the viruses that occurs in the complex of this antigenic peptide of virus occurs in the complex to MHC1 molecule. This molecule is recognized by CD8 receptor of the killer cell, and with this recognition becomes legitimate. It means CD8 receptor confirms that MHC1 receptor of target cell is self. A TCR receptor interacts specifically to the antigenic peptide. Result is beginning of the attack. Granules with the enzymes and perforins migrate to the boundary between target cell and space between target cells and cytotoxic cells. Perforins form perforations there, red enzymes enter inside of the cell, destroying structures of the cell. This is a last slide. Apoptosis of the tumor cell, or virus infected cell, or bacteria infected cell, or cell infected by fungi or protozoa, means apoptosis of cell carrying inside the intracellular parasites and destroying of the cell may be caused by what? Take a look to the left part of the slide. Here you see cytotoxic lymphocyte with the granules with perforins and enzymes. Cytotoxic lymphocyte by the T cell receptor recognizes antigenic peptide of the parasite presented by the MHC1 molecule. MHC molecule additionally is recognized by binding to this molecule of the CD8 receptor. CD8 receptor shown as with this green curved molecule. After that, perforin enzyme attack is happened and cell is lysed. In the upper part, you see the same recognition of antigenic peptide by the T cell receptor of the cytotoxic T lymphocyte. But result is fast ligand molecules of the antigenic peptide on the surface of the cell becomes polymerized and react to the many fast receptors on the local zone of the target cell. But this fast ligand fast interaction causes generation of signal causing apoptosis and self-damaging of the cell. And on the right part, you see cytotoxic lymphocyte that recognizes target cell, and after this target cell recognition secretes tumor necrosis factor. Tumor necrosis factor binds to the tumor necrosis factor R1 receptor of the target cell, and this signaling also causes apoptosis of the target cell without elimination of target cell. Now, today I try to demonstrate results of the endogenous presentation and exogenous presentation of the antigens to the CD4 and CD8 cells. Well, lecture is finished. Thank you for coming. Goodbye.